So Mendel went further and discovered that you could predict what the offspring were going to look like given information from the parents. If he knew the parents in the P generation were purebred, so he knew which alleles that they had, he could predict from the F1 to the F2 generation how many offspring were going to show the purple petal color and how many were going to show the white petal color. And this happened to be true in his experiments. So basically Mendel did a Punnett Square analysis, highlighting the different combinations that could occur in the F2 generation. In a Punnett Square, each square represents a probable product after fertilization has occurred. So I want to go ahead and highlight here that we have one parent from the F1 generation having that heterozygous allele combination, one uppercase, one lowercase, and another parent from the F1 generation showing that heterozygous, one uppercase, one lowercase, or one purple color and one white color, if you will. So crossing the two F1s together, running a Punnett and the F2, you could see how the different offspring resulted in the different squares. The possibility of getting this combination, we see a homozygous purple petal color offspring, seeing this heterozygous purple color offspring is another combination. We can see that again in this bottom one, a heterozygous combination. And then I can see this homozygous white color result. And so running this Punnett can give us this information. I want to show you guys how you run a Punnett analysis. Okay, so taking all my F1 generation, remember all the F1 generations in the previous example that I'm using is heterozygous, one uppercase allele, that's the purple petal color, shown by this uppercase P, one lowercase p, showing me the white petal color. This is a heterozygous and a visual, two alleles. So my F2 generation, I'm going to go ahead and take one parent from the F1, another parent from the F1 as I described, sperm fertilizing with egg to show us the different combinations that are possible. So how do you do this? To set up your Punnett, I want you to draw 4 boxes as so, and each of these alleles are going to combine into now 2 alleles to form an offspring, one from the top, one from the left side. Before I show you this, if you're wondering if male needs to go on the horizontal and if female needs to go on the vertical, it doesn't matter if I would have switched them. I'm going to get the same results. That's more than okay. Usually you will see the male combination shown on the vertical axis right here, and usually you'll see the female on the left. You don't have to do this. You're going to get the same results. Okay, so after drawing your 4 boxes, you go ahead and bring down those first 2 alleles in that top left box. So I see that the allele coming from the male F1 plant is an uppercase P, or purple, just uppercase P for all simplicity. And then from the female side, the left side, we see that it is an uppercase P that's going to contribute to that box. That is how you get the homozygous to uppercase P offspring in this case, and we can see that that is going to be the purple color. Remember that uppercase P coded for that dominant purple allele, so this plant will appear as purple. Now doing the same thing to those other alleles, now we have that lowercase p being contributed to that top right box from the male parent, and we still have that uppercase P that we're working with from the female parent, that left side allele, top version, and those 2 alleles will be written as such, one uppercase P, one lowercase p, one from each parent. I want you guys to appreciate at this point that you are going to write the uppercase allele before you write the lowercase allele. We usually show the dominant allele, whether it's inherited from the male or female, that doesn't matter. The dominant allele, uppercase letter, is going to be written prior to the lowercase letter. So here's a heterozygous offspring inheriting those two alleles, but this offspring, because they have the dominant allele encoding for purple color, it's going to show as purple, just like the one in the other box. It's different though because it's heterozygous. It's not homozygous for purple, it's heterozygous, having that white allele being masked by the purple dominant allele. Continuing on with our Punnett, now we have that bottom left box that we're going to work with. Let's go ahead and bring down those two alleles from each respective parent. We get another heterozygous offspring that will also show the purple color as previously described. Our last offspring, bring down those two alleles from each respective parent. Looks like we're bringing down two lowercase p recessive alleles. So that offspring is going to be homozygous for those two lowercase p alleles. I call this homozygous recessive because it has those two lowercase p alleles. And this offspring will show as white because there's no dominant allele to mask that white recessive allele. So now you finally see that white trait being shown. A Punnett Square is really showing you guys the possibilities that you're gonna get in your generation. What offspring, what's the chance that that offspring is going to show that purple color? What's the chance that it's going to show that white color? So Mendel understood that genetic crosses obey these rules, these probabilities, and it's really the same rules that perhaps you guys have learned when you toss a coin, you roll the dice. What's the likelihood that you're going to get heads or tails? What's the likelihood that you're going to get the number 6, number 5? This is all probability analysis. And so that's what's really going on with your Punnett. We are doing 1, 2, 3, 4 boxes here in this Punnett analysis. So 1 out of 4 boxes, that's a 25% probability that you're going to get this combination. This is another 25% probability that you're going to get this combination in this box, and so forth. It's important to bring new terms now. You might have noticed in this previous Punnett that you have a ratio. You've seen that quite beautifully where we have 3 purple flowers and 1 white flower. This is called a phenotypic ratio. It's called a phenotypic ratio because it's telling me the ratio of phenotype. What I mean by phenotype is the actual physical appearance of that organism. In this case, purple, 3 here, or white, 1. That's what I'm actually seeing. Sometimes we don't see it with the naked eye. Sometimes we can be talking about an internal protein for the phenotype, but it's the actual physical appearance that we see. Genotype is going to be different from phenotype in that genotype, we're talking about the genetic makeup. We're talking about the alleles that that organism has. So I can actually see your alleles. I can figure out what your alleles are based on your phenotype. Vice versa is true, and I'm going to teach you guys this. It's a different ratio from phenotype when we're looking at genotype. Here in this example, it's a 1 to 2 to 1 ratio. Why? Well, just looking at the genotype here I have a homozygous dominant genotype because I have 2 uppercase P alleles. Here I have a heterozygous genotype. Here's another heterozygous genotype, that's the same. And I have a homozygous recessive genotype here, those 2 lowercase p alleles. So that's going to be a 1 to 2 to 1 ratio. You can see that quite beautifully with that genotypic analysis in this Punnett.